Okay, welcome to Lecture 3. We're going to continue some work on binomial probabilities, and we're also going to discuss sex chromosomes. A quick note on our project, it will be due in class on this Thursday, but just a reminder, it's going to be a draft version of you're having some problems with it. We'll be able to discuss this during the office hours this week, as well as during the class time as well on Tuesday. One critical thing, however, is that if you're going to sort of dive into this and you're confused, it's really, really important that you understand how, for example, this for loop works. All right. Now, I provided the machinery for the while loop that would actually count how many attempts is made before you get a mutation, and then you have to keep track of that. So you need to think about how you're going to put something maybe inside this for loop. But to understand this for loop, you're going to have to mess around with it, right? So you should understand, why it does what it does when you spit out histogram. And the other thing that's important also is that variables can be named anything, right? So it doesn't have to be Z, or it doesn't have to be I. You could call it J or LMNOP or annoying underscore variable, whatever you want. Okay, so we'll work on that on Tuesday, hopefully, if we have time. Okay. So when we talk about binomial probabilities, the generalized framework that we've been discussing binomial probabilities is by asking this question: what is the probability of X events in N trials, each with probability P? And by X events, you could call them sometimes successes, but what we're really talking about is the number of focal events that we're interested in among a bunch of attempts, or N attempts. So for example, like if you have 5 plants and you're interested in if 2 of them are going to produce wrinkled peas, X would be 2 events, wrinkled peas, and 5 total plants would be N trials, and then you'd have those probabilities predicted by Mendel. You can also ask, from the simplest perspective, if you flip a coin 10 times, what is the probability of 10 heads in a row? In a family of 10 children, what is the probability that there are 3 boys? We provided that framework in the last lecture. In a cross between members of the F1 generation, was the probability that all 20 collected seeds are round. And then this is why we work on this, is because you can ask questions and answer questions like this. If a mother and father both carry a mutation for cystic fibrosis, what is the probability that none of their 3 children will get the disease? Okay, now to think about binomial probabilities, we developed a framework in which we basically were able to figure out what these probabilities are, and it's worth thinking about, what's your intuition for which one of these is more common? In a family of 20 children, would you expect it to be more common that there are 12 boys and 8 girls? Or in a family of 3 children, would you expect them all to be boys? So it's worth giving let yourself think about which one you think is more likely, and then we'll go onto the board right now and actually walk through how you would do that. So we have 20 and 12 for the first one, all right? So I'm going to switch over to on the board. And what we had for doing these types of problems is we calculated probabilities with this framework, where we counted the number of ways any particular arrangement was possible, and then we counted the probability of any particular arrangement. So in this particular case, for the case where I'm going to write this down, we have boys 12 and then girls 8. All right, that was one of the questions I was asking about. That was scenario 1, or A. And scenario 2, where you actually just have 3 boys in a row, right? Boys 3. All right. Now, this one's pretty straightforward. What's the probability that you have 3 boys in a row? We said it was 1/2 to the third, right? So the probability of a boy and another boy and another boy, you multiply those things out. But this is the one that we wanted to remind you how you would actually calculate that. So I will just remind you that the way you count the ways for that to be, one way is that the first 12 are boys and then the next 8 are girls. Or you could be like 2 boys and then a girl and 2 boys. There's a lot of ways to count that. And we actually can walk through that by saying that the number of trials, N, is equal to, what do you think it is? It's 20, right? And then X is equal to, well, that's the number of boys that we're interested in, 12, all right? And so I will erase this right here, and then I'll just actually write the equation. And we'll plug that in. All right. It's n factorial over, in this particular case, x factorial, n minus x factorial. So in this particular case, what we're going to plug in for n is 20 factorial over, right, 12 factorial, right, and then 20 minus 12, that's the number of girls, and that's factorial, right? And so that's the number of ways right there. So the number of ways for that to be arranged. All right, and then the probability of any particular arrangement, well, you could just pick the first one. They're all the same, right? Because boys and girl probabilities are 50-50. So it would be, 12 boys in a row. You could say 1/2 to the 12th. And then you have 8 girls in a row after that. So 1/2 to the 8th. But that would just be 1/2 to the 20th. Right? And so, that's a small number. That's a pretty big number. And let's go figure out what those probabilities are. You can do the calculation yourself in your spare time. But if we go straight to here, all right, those are the numbers that I calculated. All right. So in a family of 20 children, that probability becomes 0.12013 that there are 12 boys. And in a family of 3 children, all are boys, is 0.125. So it's actually a slightly greater probability that there would be all boys among a cohort of 3 than 12 boys and 8 girls. Now, to be clear, we're talking about exactly 12 boys, exactly 8 girls in the family of 20 children. Okay. Now, up until now, we've been working in a framework where we're talking about boys and girls, or you could talk about coin flips or whatever. But we've been saying that basically the probability of the two events is 50-50, right? But that's not always going to be the case. So what if it isn't a 50-50 ratio? In the cases in which we might have something different, where if we, for example, self a plant, big W, little w, and make sure you can walk through the Punnett square for this. And then there's 2 phenotypes there, right? So you can be round or wrinkled, right? And the way that you're round is if you end up with progeny that are big W, or big W, little w. And then to be wrinkled, you end up with the 2 alleles that are recessive for the wrinkled phenotype. And when that happens, the probability of round is 0.75, and the probability of wrinkled is 0.25. And we want to ask this question, right? In a cohort of 5 progeny, what is the probability of obtaining exactly 2 wrinkled? So what we need to do is we need to count the probability of any particular way for there to be exactly 2 wrinkled. And you could say, oh, the first 2 are wrinkled, and then the last 3 are round. But the generalized framework for these types of things is that, again, you talk about X events and N trials. But here now, instead of 0.5 always being, the case for the probability, we give a value of P for the probability of success and a probability of 2, which is 1 minus P, as the probability of failure. All right, and, success or failure is arbitrarily defined. But the way that once you have this framework, then you basically have this equation. All right, so on the left side here, it's the number of ways, which we've been talking about before, but now instead of 0.5, you actually just have P, all right? And it's P to the X, right? It's basically the probability that this event happens this many times, X times. So, in one particular instance where, the event of wrinkled happens 1, 2, 3 in a row, it would be, whatever P is, which is actually 0.25 for wrinkled, to the 3rd power, right? And then it would be Q, which is 1 minus P, to the remaining, N minus X, right? Just remember, if it's, 5 progeny and we're talking about 2, then in this particular case, N minus X would be 3. So it would be 0.75 to the 3rd power. Okay. So this is an equation that you'll need to basically remember. I mean, obviously you'll have it since everything is going to be open book this semester, but you should just basically memorize it anyway because you're going to want to be familiar with it. Okay, so let's walk through this particular framework here. So if we have this particular case where we have these two purebred strains, which are big W, and little w, and you cross those, and now you self those, all right? So you're collecting, 1, 2, 3, 4, 5. What is the probability that two of them are wrinkled? So you want to use that framework that we had before, and I'll wait a moment and let you sort of think about it for a minute, and then I'll tell you what the answer is. I should play music during these times. Do 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 do. Okay, you don't want me to do that. Actually, I don't want me to do that. Okay, so the correct answer is D in this particular case, and that's because there's 5 progeny, so that's n, so 5 factorial, and the 2 being wrinkled, 2 factorial, 3 factorial, and then the probability that 2 of them end up being wrinkled, maybe you just say the first 2, if you want to think of one particular arrangement, is 1/4 to the 2, and then the remaining ones are not wrinkled, and the probability that the remaining ones, if there's a particular cohort, as with 5, is 3/4 to the 3, and that's about 0.264. Okay, and you can leave these unreduced if I say, you don't need to provide the exact number. Okay, now the next thing that is something that's worth thinking about is that this is all assuming that everything is a-okay with Mendel. Now I want to walk through a little bit of a funky problem in which actually sometimes pollen viability can play a role in predicting and perturbing ratios that we might be predicted under Mendel. All right, so let's think about this one here. What happens if half of all the little w pollen die before fertilization? All right, so what that means is, something's wrong with the little w pollen in those F1s. All right, it doesn't matter here. Half could die, but all of the pollen will still be little w. But in this particular case, big W is going to be more represented through the outcome of meiosis, and there's many reasons why this could be. So let's walk through how this would work, and we're going to do that on the board. So half of all the pollen die in fertilization. Oh. Okay, here we go. All right. So we're going to go onto the board here, and we want to have our Punnett square as before. And typically I'm showing the pollen granules, the genotypes of the pollen granules. I've usually been showing them up on top. It doesn't matter, you can flip the order here. But this would be the pollen genotypes. And then these are going to be the ovule genotypes. Okay, so this would be, remember, this is big W, little w, and then I'll use sort of this like circle with an arrow to indicate selfing, right? So the pollen genotypes are big W, little w. This problem with the little w pollen isn't going to change just the genotypes, but what is it going to change? It's going to change the probabilities. Now it's just pollen, right? So here the probability, is equal to 0.5, or let's say a half, for big W ovules and 1/2 for little w ovules genotypes. Okay, but what about it here? Now you might say like, okay, half. So let's just like put 1/2 and 1/2 and just say, okay, let's not worry about that. And then say, okay, well now we're going to think about how we're going to need to adjust these probabilities. Now you might say to yourself, okay, well there's half viability for these ones. Okay, so instead of a half, we're just going to erase that and we're just going to say 1/4. Now there's a problem with that. So we're saying here that half of the pollen have big W genotype, and then 1/4 of the pollen have little w genotype. What's the problem there? Well, those two probabilities aren't adding up to 1. Something's funky there. Where's that other 1/4? All right, where's this like sort of ghost pollen? All right, there's what you need to do is you need to basically adjust these probabilities so that this one is half of that one, but then they still have to add to 1, right? And 1/2 plus 1/4 don't add to 1. And one one way you can do that, I have some space down below here. I'm going to erase this. Is you can just say, okay, well, let me just say that the probability of a big W, right, is equal to, let's say it's X, right? I mean, I don't know what it is, but I do know that there's a relationship because the probability of getting a little w, what's that going to be equal to? Well, it's going to be half of X, right? Because this probability, I don't know what it is. I know it's not a half and a quarter, right? But what I'm going to do is I'm going to solve for X, and then I'm going to kind of solve relative to each other. So probability of little w, in terms of X, what is it? Well, it's half as much, so it's 1/2 X. Right? Now, you might be able to like mentally just sort of get your head to sort of stare at this for a minute and be like, well, I think I know what it should be, and it adds to 1, and it's half as much. But this is sort of an algebraic way of doing this. Well, what do these two things have to add up to? Well, X, which is the probability of getting that one, plus 1/2 of X, all right, is equal to, well, it better add up to 1, right? All right, so this is 1x, this is 1.5x, so let's just call it 3/2 x is equal to 1. And remember, x is this probability here, and so then x, see if we still have a little bit of screen. I'll do it over here. So you divide by 3/2 or multiply by 2/3, so then x is going to be equal to 2/3. Okay, let's see if that makes sense. So we're saying that instead of half, this is now 2/3. All right. Okay, well, if that's 2/3, then this has to be half as much as that. So what would that be? Well, that would be 1/3, right? And you can kind of see, like, oh, this is a ratio of 2 to 1 here, 2/3 to 1/3, and what is it? It satisfies the condition of the probabilities are adding up to 1. It's a little bit tricky, but that's the way to do it. But you could sort of stare at it and be like, okay, it's 2 to 1, and they have to add up to 1, and you can kind of get your head around it that way if you want. All right, these two, of course, are adding up to 1. Okay, so 2/3, 1/3. This is why I like to actually make you, in the last class, think about these probabilities in a Punnett square as basically fertilization probabilities, not just like 1/4, 1/4, 1/4, 1/4, because the probabilities don't always have to be half, 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 half for the gamete. Mendel usually suggests that they are, but that's not always going to be the case. So let's walk through and find out the probabilities of the different genotypes. Okay, well, this is going to be the big W genotype. It's going to be the big W, little w genotype. This is going to be, well, the same thing, but we'll just call it little w, big W, just for posterity, and then little w, little w. Okay, so what's the probability of this? Well, this is now 2/3 times 1/2, all right, which is equal to 2/6, right, or 1/3. All right, so that's clearly not Mendel right now, right? Okay, so then what about, let's do this one here. So 1/2 times 1/3, right? 1/2 times 1/3. I guess you could do it 1/3 times 1/2, whatever your order, doesn't matter. But anyway, this would be 1/6 now. Okay, and then this is 2/3 times 1/2 equals 1/3 now. Okay. And what about this one? 1/2 times 1/3 is equal to 1/6. Okay, now that's funky, right? But if you have different viabilities, you can have these types of things. And it doesn't necessarily just have to happen with pollen. It could happen with ovules. It could happen with both of them. So you'd have to adjust it in the same kind of ways on this side and this side. It's worth checking to make sure that all of those add up to 1. 1/3 plus 1/3, 2/3, and then these together make that 1/3, and so the whole thing adds up to 1. So that was the, let me make sure I actually answered the question. It's always a good rule of thumb. Okay, do we get a 3-to-1 ratio in the F2? Certainly not. Okay, so we're going to keep on marching through thinking now we're going to go back to cytology for a little bit, okay, away from probabilities. And we're going to talk about, and remember, I actually, when I introduced mitosis and meiosis, I described this interesting observation that the cytologists had made, that it seemed that the nucleus was important for inheritance. And it also seemed that chromosomes were important for inheritance because the chromosomes were inside the nucleus. Cytologists could see them, and they could see that they tended to split and segregate in even proportions among daughter cells, either in meiosis or mitosis. And so there was a concept that, maybe chromosomes carry genes, or maybe chromosomes actually are genes, like maybe each chromosome is a gene. But there was another school of thought, which you could see is reasonable, is that maybe it had nothing to do with genes. Maybe it was just the scaffold that the nucleus was structured upon, and actually genetic information wasn't on the chromosomes themselves. And so we're going to talk right now about how do we know, or how do geneticists figure out that genes resided on chromosomes. And the way that we know that is actually through what are known as sex chromosomes. And Nettie Stevens, a woman, was one of the people who was the first to really find this with her mentor Edmund Beecher Wilson. So you should remember her as an important woman who contributed to the early history of genetics. You don't necessarily learn about her in school, but you should. She was the woman who started squashing grasshoppers and basically looking at their cells. And what she and her mentor found is that there are differences in the way that the chromosomes look in female grasshoppers versus male grasshoppers. And this led to this observation that females have two pairs of chromosomes that look similar, but males in this case have what is called a Y chromosome. Okay, and the Y chromosome is a smaller version, and in this line drawing here, so the photographs weren't always used in the literature at this time. Photography was hard, especially through a microscope. But you can see then that in cases there with these chromosomal squashes, oftentimes the males would show this sort of small little aberrant chromosome called a Y, right? And you can see a little Y chromosome right there as well. Now, it's interesting to think about, when you make the observation that males have the XY combination and females have an XX combination, you might say, oh, okay, I think that the sexes are determined by the chromosomes. That might be a logical inference, but it's not a perfectly good inference because it could be the other way around, right? You could have a scenario in which sex is determined through some other mechanism, right? Maybe it's temperature, maybe it's not genetic at all, who knows, right? So maybe it's the hormones of the mother that, are placed in the egg or something like that. But, it could be anything that determines sex. And then once the sex is determined, then the sex determination is upstream of the way the chromosomes look. And maybe males, for whatever reason, need to degenerate one of the X chromosomes into a smaller Y chromosome, right? So in this particular case, it's not the chromosomes determining the sex, it's the sex determining the chromosomes. And so how do you figure that out? That actually played a role. Figuring that out was what was critical for us to identify that genes reside on chromosomes. And the first evidence for a relationship between genes and sex was found in the particular case of the Drosophila melanogaster. And Drosophila melanogaster has been a common workhorse for genetics for more than 100 years now. And this was work out of the Morgan lab in the early part of last century from Columbia in New York. Just introducing you here to male Drosophila on the left and female Drosophila on the right. And then you can see that normally these flies have red eyes that look sort of like a brick color. But mutations can be found all the time. If you just stare at it in thousands of Drosophila, you'll find mutations. I've found them in my lab. And one of the most well-known mutations is the mutation that causes the red eyes to become white, and this is a mutation called white. All right, and so if you're a geneticist, if you were to find in a vial, you're staring at your vials of Drosophila. I'll actually bring one over in a minute. Here, I have one over here. I have a vial of Drosophila right here. I can show to you because I'm in my lab. And give me a second. Gotta go to Zoom now. All right, there's my 81 strain of Drosophila, which actually doesn't have white eyes. But anyway, going back to this, so you can stare at these fruit flies, till kingdom come, and you'll find mutations in them. Now, if you're a biochemist, and this is me speaking a little cutely here, but if you're a biochemist and you find a white-eyed Drosophila, one of the things you might do is grind up that Drosophila and see what might be different about red pigments versus white pigments, right? But Thomas Hunt Morgan was not a biochemist. Thomas Morgan was a geneticist, all right? And if you consult Mendel's ideas, if you find a white-eyed male, what do? Well, if you're a geneticist, what you do is you cross it with something. Because what you want to be able to do is establish a line of Drosophila that has that white mutation, that white eye. Now, you don't know what's going to happen, right? You don't know what's going to happen with this white mutation, and so you might cross it. And in this particular case, you cross it, and then in the next generation, actually, all the progeny have red eyes. And I'll show you why that is in a moment. So then what do you do with that? Okay, well, if all the progeny have red eyes, you cross them. All right, you do whatever you can to see if the white eye will pop back up. All right, so there's a couple key points that I need to explain before we actually discuss how inheritance works in the case of sex chromosomes. So the first thing I wanted to say is actually, like humans, in fact, in Drosophila, males are XY and females are XX. Now, just to be clear, that's not because we share a sex determination system with Drosophila. All right, so those are independent events that originated. So it just so happened that Drosophila, actually Diptera flies, millions of years ago evolved an XY system. And then actually, basically, all mammals share an XY system, but those are independent events. So, but in both, humans and in Drosophila, the Y chromosome basically has no genes on it. It has a couple. I want to be clear. It has a few, but for the purposes of this class, we can just say that there's basically no genetic information on the Y chromosome. And that is then, therefore, why we say that a male Drosophila, which is XY, is what we call hemizygous for the X. All right, you can't be homozygous because you only have one X. You can't be heterozygous because you only have one X. But hemi means half, and so you're half zygous for the X. All right, so any gene that you have on there, you're not homozygous for it because you don't have two copies. You're hemizygous for it. And this is important and relates to what we talked about in the first class. If a fly has a single normal copy of the white gene, now when I say a single normal copy of the white gene, the mutant is what makes white eyes. So the wild type version, the normal copy, has red eyes. All right, and that happens for males and females, right? So if a male is hemizygous for a wild type, he has red eyes. But if he's hemizygous for the mutant, he has white eyes. If a female is homozygous for the wild type allele of white, she has red eyes. If she's heterozygous for the wild type allele, she has red eyes. And she's homozygous, she has white eyes. And so this is because the white gene is haplosufficient. So having one good copy is enough. All right, and the mutant allele is recessive. Okay, so we're going back. So you do some genetics, you have a white-eyed male, what do? You cross it with something. And I said the first cross was a little bit weird, but then some further crossing happened. When I say weird, it basically, Thomas Hunt Morgan didn't see the white-eyed in the next generation. But if you're a geneticist and you have a generation where you don't see the white-eye, you're going to take those progeny and keep on crossing them. Okay, so in this particular case, this was the first cross that was done. You can actually say what are the progeny ratios here for a female that's breeding true, all right, meaning she's wild type, and the male that has the white eyes. All right, so we're going to do this on the board now. Okay, so the way we do this is we set up a Punnett square, just as before, right? And then usually, we're not talking pollen here, but we're talking sperm. We're talking male gametes. So male gametes. And then the female gametes over here. And, my notation might be a little different than your notation. But what I do here is that I put an X chromosome, right? But it turns out that white is on that X chromosome, all right? So in this particular case, we have an X chromosome, and I'm going to put a W plus on there, all right? Oh, excuse me, a W minus. He has white eyes. Sorry about that. So he's mutant, the male, so he has white minus there. And then the other chromosome, well, does he have another X chromosome? No, he has a Y chromosome. All right, Y. All right. And do you put W plus or W minus there? No, because there's no white gene on the Y chromosome. Now the female has, she's wild type, so we do an X here, an X here, and we put a white there, and then we just do plus, all right? And then the white gene, and we do plus, all right? So she has red eyes, and he has white eyes. All right, so how do we walk through this, all right? The way we walk through this is we're going to actually watch two things happen, all right? We're going to watch sex determination happen, which depends on the Y chromosome, and we're going to map the nature of the genotype with respect to white also onto those individuals, all right? So he can transmit either a Y sperm or an X sperm. Let's do this column here first. So he transmits a Y sperm here and a Y sperm to this column, all right? And then he transmits the white mutant here, so white mutant, all right? And then he transmits it on the X chromosome, white mutant, all right? Now, what gametes do these females transmit? Well, they're homozygous, right? So they're going to transmit only the X chromosomes that have the wild type. So we put just the same XW plus now, XW plus, always the same here, XW plus, and then the X with another W plus, right? Because she's homozygous, so she could. All right, so now we're going to look at these now. The first thing that we want, you could do it however you want, but the way I would do it is the first thing we do is we look at the sexes, okay? And so this is all 50-50, one half, nothing weird about distortion of the ratio. So this is going to be 1/4, 1/4, 1/4, and 1/4. All right. So, and because of this, 1/4 plus 1/4, these ones, right? These are the males, right? And the reason we know they're males is because they have a Y chromosome. This is what's making this one male, and this is what's making that one male. So these are the males, right? And half of them will be male, right? And then X, 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 X. So the other ones are the female, right? And so it's a 50-50 ratio, just as you expect. And so, with sex determination through chromosomes allows a 50-50 sex ratio to be maintained in a population. All right. Now, we have females here and males here. And then what is the phenotype now? Now, the phenotype here, this is white plus over Y, and this is white plus over Y. It's haplosufficient. So what eye color will they have? They're going to have red eyes. So these ones, red eyes. Okay, I should put it down here, red eyes. Can't tell if I'm chopped. No, red eyes. All right, so all the males have red eyes. What about the females? Now, the females are heterozygous, right? They have one mutant allele. They have the mutant allele that they inherited from the X chromosome of their dad. Sons collect the Y chromosome from the dad. Daughters collect an X chromosome from their dad. Okay. So they're heterozygous. They have a mutant allele here, but they have plus, right? And the white mutant allele is recessive, right? So these are also going to have red eyes. And so the prediction, this is what I was saying before, is that like, oh, that was probably quite terrifying. Like you have this white-eyed male, you cross it with obviously a wild type because you don't have any other mutant females around, and now it's gone, right? Because you have red eyes among the females and you have red eyes among the males. All right, so what do you do next? So the next thing that you do is you might do another cross, right? As a matter of fact, what you can do is you can do what is called a back cross. So you have this original male that you found in a vial here, right? You're not going to throw him away, right? You're going to take the female progeny and you're going to cross again to this white-eyed male. Now, why would you do that? So you're keeping him across 2 generations. This is something you can do with fruit flies. Obviously, you can't do these types of stuff in humans, but you would do this with, in this particular case, the one white-eyed male you have, you want to mate him with the females that you've collected, right? So how do we walk through that? So let's walk through that now. We'll do again a Punnett square, but with these different genotypes, but with sex, also sex linkage. Okay, so I'm going to go back to the board, and now we have a heterozygous female, right? Because we just talked about how all the females of that cross are heterozygous. So we'll go back to our Punnett square. The males, well, he has white eyes. And white minus and a Y chromosome. And then we said that those females were heterozygous, so one of the chromosomes will be white plus, and another one of the chromosomes will be white minus. So before, it looked like maybe this was only happening in males. It's an interesting observation. Like, wait, I only found a male and it seemed to go away. Can I have this phenotype show up in females as well? Well, it's something you could ask. So again, Y chromosome here. I'm going to move a little bit more quickly here. Y chromosome here. X with a white mutant allele here. X with a white mutant allele here. Right? Now, this gamete is wild type because the mom is heterozygous. So this would be X white plus. And then the same logic for here, right? The daughters, which have 2 X chromosomes, white plus. And then this would be, okay, X white minus and X white minus. And this is interesting now because what this predicts is, these will be the males and these will be the females. These females and these males have red eyes, right? Because they have one of the plus alleles. But these females are homozygous with a mutant allele. Right, so they're going to have white eyes. So it's going to be a 1-to-1 ratio among the females of red to white. And the same thing is going to be true here, right? A 1-to-1 ratio of red to white. Right. So you should be familiar. You could be able to, walk through both sex and genotypes in Punnett squares that way. And we'll get to this more when we talk about pedigrees. Okay, there's a couple interesting things to think about before we end this lecture. Human females and Drosophila females are XX, and human males and Drosophila males are XY. So, in Drosophila and humans, we say that males are what are called heterogametic, and hetero means different, and gamete, means gamete. So it means that the gametes have 2 different genotypes with respect to sex chromosomes, right? But homogametic females means that females, their gametes with respect to the chromosomes are the same, right? All of their gametes have just an X chromosome, right? But not all organisms have heterogametic males and homogametic females. In birds and in butterflies, it can be switched around, and we call those ZW and ZZ systems. So in this particular case, it's the W here that is a sort of like analogous to the Y chromosome, right? Because the males are ZZ and the females are ZW. So the W is analogous to the Y, but the females are ZW, so the females are heterogametic and the males are homogametic. And so that's something that's important because, the patterns of how you would watch phenotypes through a Punnett square would be different, for example, with birds or with butterflies. Now, this is important. Remember earlier on, just in this lecture, I talked about how it wasn't clear 100% sure if genes were on chromosomes. So how do you actually prove that genes are on chromosomes? Now, the way that this was proven was using the white mutant alleles. Right? Now, the way that this was done is one can make a prediction then. If, in fact, genes reside on chromosomes, then that would mean that these white alleles would reside on chromosomes, all right? And we know they do obviously reside on chromosomes, but we didn't know it at the time. So one can make a prediction. If chromosomes segregate improperly, and by what you would see with chromosomes segregating improperly is, that would be seen cytologically, right? Like in a microscope, right? If chromosomes segregate improperly, then the genes will also segregate improperly because the genes reside on the chromosome. So when something gets messed up with the chromosomes moving around in meiosis, then this would also lead to something weird with respect to the way that the phenotypes are connected to the genotype. And so how do we actually make chromosomes segregate improperly? Well, it turns out nature does this for us. There's meiotic problems that can be called meiotic non-disjunction. All right, meiotic non-disjunction occurs when the chromosomes do not segregate properly in meiosis. Now, we've already said that for a particular pattern of sex chromosomes, in this particular case, males being wild type and females being mutant, you can walk through the Punnett square. We didn't do this particular one, but if you can walk through the Punnett square, the females will all be expected to have red eyes because the males are going to transmit the wild type allele to his daughters, right? And he'll transmit the Y to sons. Now, the females are going to transmit the X to both their sons and daughters, but the sons only have a Y chromosome, so this predicts then that the red-eyed females should arise and that the F1 males should all have white eyes. Okay, but that's not always what happens. Sometimes something weird happens in which you get a white-eyed female or you can get a red-eyed male. So the question is okay, something's gone wrong weird genetically. Did chromosomes missegregate in that process? And so you can walk through how this non-disjunction works. In this particular case, the non-disjunction will have happened in females. You can walk through different examples, but this would be the way it worked when these experiments were done. Non-disjunction sometimes happen in females, and when non-disjunction occurs, instead of one chromosome being in one gamete and the other chromosome being in the other gamete, here you can see that there are 2 chromosomes in one gamete and 0 in this one. Right? Now, this is a problem. All right, this is actually called a diplogamete, and this is called a nullogamete. Now, these diplogametes here, in this sort of funkadelic Punnett square here, will be transmitted, and then this will be united with a sperm that's white plus. All right, and this pair of white chromosomes will be paired with a Y chromosome here. Okay, and this nullo here, all right, will be fertilized by a white plus sperm, all right, and then this is the worst scenario here because here no X chromosome is transmitted and then there's a Y chromosome. That's what this sort of weird little bent chromosome is. If an organism just has a Y chromosome and no X, remember there's a lot of critical genes on the X. This individual will be dead. As a matter of fact. These individuals will be dead, and actually these usually die as well, all right? So what you observe here is these are the outcomes, right? These are the most common outcomes from non-disjunction, and these are the ones that are like, oh, there's these weird white-eyed females that I don't predict, or these weird red-eyed males. Now these ones are sterile, but if you look, and you would probably mostly ignore these ones here because most of them are dead. But if you look at those red-eyed males, which you did not predict, and you looked at them cytologically, you would find there's no Y chromosome. And that means that they did not likely inherit, or they wouldn't have inherited an X chromosome from their mom. And again, in this particular case here, we have these white-eyed females. Oh, they also do have a Y chromosome. Now, how does a female have a Y chromosome, right? Well, it turns out it turns out that sex determination in Drosophila just depends on the number of X chromosomes that the individuals have. So having an extra Y chromosome doesn't make those ones female. Just the two X's make them female, and the one X makes males males. So it's a little different in humans. But the point here is, when there is this sort of weird, aberrant, white-eyed female, a Y chromosome could be seen cytologically. And that basically showed that faulty genetic inheritance with respect to sex and eye color coincides with faulty chromosomal inheritance, and that was what proved that genes reside on chromosomes.